In Chapter 13, we'll be talking about biodiversity, and we'll also be spending some time talking about how biodiversity is important to humans. Interestingly, sometimes biodiversity is extremely important to humans from a financial perspective. One of the most common examples that's discussed regarding this aspect of the value of biodiversity is are the prokaryotic organisms, archaeans, and bacteria that are found in the hot springs in Yellowstone National Park. And so you'll recall PCR from a few chapters earlier in our class, the polymerase chain reaction. And you remember that in PCR, the DNA polymerase that actually performs the DNA replication is not the DNA polymerase from human cells, but it's a DNA polymerase from one of these prokaryotic organisms that are adapted to life at extremely high temperatures. And so this is an example of a case in which an organism that no one would have ever guessed, say 30 years ago, would be of any value to humans, has been worth a tremendous amount of money. Hoffmann La Roche, which is the biotech company who first came up with a PCR that Kary Mullis, as you recall, was associated with. It's worth $100 million it was worth $100 million a year to them every year until the patent expired in 1999. And so now it's worth more than that, undoubtedly, but to a number of different companies. So we want to spend some time talking about biodiversity in this chapter and the value. In some cases, we'll talk about the value of biodiversity to humans. So biodiversity is the variety that we find within and among living species. There's about 1.4 to 1.8 million species that are known to science on this earth, but there are lots and lots of species that have not been identified, that we believe are out there but have not been described. We either haven't come in contact with them at all, or we simply haven't named them at this particular point. So there's somewhere between 3 and 100, and I realize that's a tremendous range, but 3 and 100 million species that exist in the world total, we think. And so it's interesting to think about it, and remember that many of these undescribed species, you may say, well, that's odd. There's animals out there we haven't actually discovered, but many of these species are fairly species that are not too noticeable. Insects actually probably make up the biggest bunch of these organisms that we haven't described yet. And then prokaryotic organisms and fungi and other microscopic or semi-microscopic organisms probably make up a lot of these undescribed species. And so I want to spend just a moment talking about how it is that we discover species and how we officially come up with a name for a species. And so interestingly, this is done when a scientist discovers an organism, writes a description of that organism, and gets that description published in a recognized journal. On the screen we see Dr. Guenter Schuster, who interestingly was my advisor when I was in college, and the top picture shows Guenter Schuster after Dr. Schuster after a tropical biology trip, which must have been a fun class, but they went, I think they were coming through Jamaica at that time, some tropical destination. There was a lot of trouble at the airport, and they ended up staying there for a long time, and he went to sleep there, and one of the students wrote a sign that is a joke there on top of on the seats that are just above him there. But Dr. Schuster was a taxonomist, and so we have one of the species that Dr. Schuster described in the lower right-hand side of our slide here. And so this is a species of crayfish, and it's actually a species of crayfish that they described in 2010, just a couple of years ago. This is a huge species, like 8 inches long. I mean, that's a really big crayfish, and it's found in the border of Tennessee and Kentucky. It's not very common at all, but it's just something that no one had ever noticed before. And so they found it, they wrote a description of it, and they published it in the Proceedings of the Biological Society of Washington. And that's how new species are officially recognized. In order for an animal to be recognized as a species, that process has to be conducted. Okay, so as we begin our discussion, then we want to talk about the classification system that we use to categorize this biodiversity. We now divide species into three domains. In other words, there are these categories that we place living organisms into, and the three biggest categories that all living things on the face of the earth fall into are referred to as domains. There are three domains: domain bacteria, domain archaea, and domain eukarya. We're going to talk about each of these briefly, but the first two domains are prokaryotic, and the last domain, as the name would imply, is eukaryotic. We'll talk about the difference between bacteria and archaea, but briefly, bacteria are more common, and they are the organisms that live on you. You realize, hopefully at this point in the class, that you have bacteria that live in your nose and your eyes and on your skin, and all around you. Bacteria are found in the soil and almost everywhere. Wherever you are right now, as you look around at the desktop or the tabletop around you or the floor, there are bacteria that are found all over those surfaces. Hopefully not harmful bacteria, but regardless, bacteria are all around us. Archaeans are a little less common. They're the same size as bacteria, even a little smaller in many cases, but they're found in fairly extreme environments, places where it's very hot or the pressure is very high or it's very salty. And so we'll talk a little bit more about them as we go forward in our discussion as well. Within the domain Eukarya, we have four kingdoms. We have Kingdom Plantae, Kingdom Animalia, Kingdom Fungi, and Kingdom Protista. Now, the other two domains don't have multiple kingdoms. In fact, Archaea doesn't have any kingdoms that are recognized at the moment, but bacteria does have a kingdom bacteria inside the kingdom. Inside the domain bacteria, which may not make much sense to you, but that kingdom still resides inside that domain, although it does seem a bit redundant. But Eukarya has four different kingdoms that are found inside it. So remember, as we start at the top and make our way down, the biggest category we have are domains, and then underneath that we have kingdoms. Nucleic acid sequences are primarily the method that's used today to classify organisms. Now, you realize this from some of our earlier discussions in previous chapters, but it's interesting to note, and it's also interesting to contrast this with the method that we used to use to classify organisms. We used to just look at organisms and visually place them into different categories based on their appearance. Today we don't do that. Instead, we sequence DNA from organisms, and we sequence DNA from very particular sequences of those organisms. And so what we need, if we're going to use DNA to classify organisms, are sequences that are found in each category of organism. So in other words, we wouldn't want to use, say, the sequence for fins to classify a bunch of organisms, because all those organisms may not have fins, right? So what we do is we use the DNA that codes for the ribosomes. All organisms, as you realize, must have ribosomes in order to survive. And so we use the DNA that codes for ribosomes and compare those sequences among various organisms to construct phylogenetic trees, such as the one that we see on the screen. You'll remember from earlier in our class that a phylogenetic tree is a reconstruction of the hypothesized evolutionary relationships between a group of organisms. Here's an example of a comparison of sequence, and that illustrates that DNA sequences are different in different organisms. So here we have long-eared sunfish on top, and we have zebra fish on the bottom, and we're comparing the sequence of a particular gene found in these fish. Now, this is not ribosomal DNA. Now, usually ribosomal DNA, as I just said, is used, but this is a different gene. This is a gene for metallothionein, which is a particular protein in these fish. This is from some work that I did a couple years ago, and so that's why I'm including it at this point. So we had the gene originally for zebrafish, here shown at the bottom. We had that sequence, and myself and my colleagues sequenced the gene for long-eared sunfish, and we compared the two, and so you can see that they're mostly similar. They're 83% similar, but there are now and then differences, such as right here. In zebrafish we have a cytosine here, and in long-eared sunfish we have a guanine. And so there are differences in the sequence, and that helps us put together a classification scheme for these fish. When we start to do this with more and more species, it helps us put together a phylogenetic tree for this group. Alright, so we want to begin by talking just a little bit about bacteria and archaeans. Remember, these are the first two domains, and both of these are prokaryotic organisms. We'll start with bacteria. Domain bacteria. The domain bacteria contains millions of different species. We're most familiar with the bacteria that cause disease. So pneumonia and tetanus and pertussis, whooping cough. And what else? All sorts of diseases are caused, tuberculosis, are caused by bacteria. But interestingly, the bacteria that cause disease are certainly not the most common type of bacteria out there. Most bacteria have no adverse effects on humans at all. But we're familiar with the ones, of course, that cause disease because they're of significance to human health. Many bacteria are decomposers. They play an important role in the environment because they decompose organisms that are dead. Bacteria do produce compounds that are useful to humans, and so you realize that antibiotics such as penicillin are produced. Actually, penicillin is produced by a fungus, isn't it? Of course, Penicillium. But other antibiotics, such as streptomycin, are produced by bacteria. Many different types of antibiotics are produced by bacteria. About half overall are produced by bacteria. Restriction enzymes, you'll remember them from our just last chapter, are also come from, perhaps that wasn't clear from our discussion there, but they do come from bacteria. You remember the plasmids and how the plasmids are cut by restriction enzymes, and then other types of DNA can be glued to, so to speak, with those sticky ends, the bacterial plasmids. Well, those restriction enzymes are found in bacteria. Fecal coliforms are an interesting type of bacteria. Remember, in your large intestine you have almost a zoo of bacteria. Perhaps a little disgusting to think about, but you have over 500 species of bacteria residing in your large intestine. And these bacteria are known as fecal coliforms. They play a beneficial role in your overall health. They help you absorb nutrients that otherwise would be difficult for you to absorb. It's been demonstrated that when they take mice and do a C-section on a mouse and raise that mouse in a sterile environment, in other words, the mouse never accumulates those bacteria in their intestine, then even if they feed the mice a diet that has plenty of nutrients, everything that they need, the mice never do that well. So fecal coliforms are significant for this reason. They help us detect pollution in water. And so when we go down to the Kentucky River, when we take we get some water from the river, we pass it through a filter, and then we take that filter and we press it down onto a petri dish that has agar on it, as we see on the right hand side of our screen. Then we incubate that plate and we let those bacteria grow, similar to what was done in your lab last week. And what we see is that the bacteria form little colonies on that plate if they're present. So we're growing the bacteria on a special type of media, a special type of agar referred to as eosin methylene blue. And this particular agar causes fecal coliforms to show up as blue. So the take-home message is this, that you filter the water, you press the filter onto that special media, and if blue colonies show up on the media, you know fecal coliforms are in the water. And this means that you have fecal contamination in the water. Interestingly, that's pretty common in the Kentucky River. In fact, from time to time it's recommended that you not swim in the Kentucky River as a result of the fecal coliforms that are present in that river. We have a fairly pervasive problem with that in the state of Kentucky. Alright, let's talk briefly about the archaeans at this point. Archaeans are superficially similar to bacteria, but their cell membrane structure differs quite a bit. And archaeans are not nearly as common, and they tend to be found in very extreme environments: high pressure, high temperature, high salt concentration. So places like the Dead Sea, the Great Salt Lake, the hot springs out in Yellowstone National Park. All of these are areas in which we would expect to find archaeans. Archaeans are more, interestingly, a very different genetically as well. In fact, it seems strange to say this, but our best science indicates that they are more closely related to eukaryotes than they are to bacteria. Moving on to eukaryotes, then we want to talk about several different kingdoms within the eukaryotes. The first of which we'll talk about are the protist, kingdom Protista. The protist are the simplest of the eukaryotes, and the protist are thought to be one of the first eukaryotic cells that originated here on Earth. The endosymbiotic theory is an interesting idea, and the endosymbiotic theory states that back when we just had prokaryotic cells, one prokaryotic cell engulfed another one, and then instead of digesting that prokaryotic cell, the second prokaryotic cell, the one that was eaten, remained alive inside the first one and started to perform some useful function inside that first prokaryotic cell. And so that's the endosymbiotic theory, and that's widely believed to explain how eukaryotic cells came into being. Modern protists resemble plants, animals, and fungi. On the bottom of our screen, on the left-hand side, we have a animal like protist, and so this particular protist is known as Giardia. It's a very small, visible to the naked eye, but just barely. It's found in all freshwater systems here in North America. So Giardia is the reason why you shouldn't drink water even from the cleanest looking spring. There may be these little organisms in there, and if you drink those organisms, they munch on the inside of your small intestine, giving you very, very bad diarrhea until you can get rid of it. In the middle slide on the bottom, we have a picture of Volvox. Volvox is an algae-like protist, and so it's a plant-like protist. It is algae. Algae falls into the protist category, and so algae, including Volvox, are all examples of plant-like protist. And then on the far right, we have a fungus-like protist. And so the slime molds are examples of protist. They're not classified as fungi. They don't meet all the characteristics of fungi, but they do appear like fungi. So interestingly, Kingdom Protista contains a wide variety of different types of organisms that are all lumped together into this category. Let's move on at this point to animals and talk a bit about Kingdom Animalia. Animals there's lots of different types of animals, but they all share a couple common features. They're all multicellular, they're all heterotrophic, and they're all mobile during at least one stage of their life. Some animals, interestingly, have two different stages of their life, and they may be sessile during one part of their life. Many ocean invertebrates remain attached to a rock during one part of their life, but they're free swimming during the other part of their life. The animal Kingdom Animalia appear to become much more diverse if we're interpreting the fossil record correctly about 530 million years ago. At this time, something called the Cambrian explosion occurred, and multicellular organisms, in particular animals, seem to proliferate very quickly. And after this point in the fossil record, we see a wide variety of animals present. And so, in the Kingdom Animalia, we see organisms like Porifera, which are sponges, the very simplest organisms that fall into Kingdom Animalia. We see the Cnidarians, things like sea anemones, which are still a fairly simple animal, but they certainly are an animal, and they're a little bit more complex than are the Poriferans. We see Phylum Platyhelminthes, which include the tapeworms. Tapeworms, we'll look at a life cycle in just a moment, but tapeworms are interesting parasites. Parasites typically have fairly complex life cycles and simple body styles, which is definitely true for the tapeworms. We have phylum Mollusca, which include things like snails and clams, and interestingly octopi, which appear fairly different than, say, snails and clams, but they all fall into this phylum. And the annelids, things like earthworms or segmented worms, also fall into kingdom Animalia. Nematodes are an interesting group of worms that are mostly very small. In fact, many of them are microscopic, but they're very abundant in the soil. Some of them are aquatic, but more of them are simply found in the soil. If you went outside and picked up a handful of soil, you would find thousands of nematodes in that soil. The arthropods are an interesting group that include things like shrimp, lobsters, crabs, crayfish, and insects most abundantly. All fall into this particular category. Arthropods have an exoskeleton. They have a skeleton found on the outside of their body and no bones on the inside of their body. Phylum Echinodermata include things like sea urchins, sand dollars, and starfish. These are animals that are fairly simple. They have just one opening that serves as the mouth and an anus, but they're fairly interesting and have interesting methods of eating. Starfish, for instance, evert their stomach and digest their food externally and pull that food back, those nutrients back into their stomach. The chordates, finally, are the animals that have backbones, and so things like duck-billed platypi, like we have on the screen, and birds and reptiles and amphibians and mammals all fall into this category of chordates. Here we have the life cycle of phylum Platyhelminthes, the tapeworm that we mentioned earlier. And so here we have the tapeworm. Let's see, where should we start? Sorry about that. Let's see. So let's start here. So the tapeworm reproduces when it jettisons. It's found inside the intestine of a human, and it jettisons a few of these sections referred to as proglottids. The proglottids contain eggs and sperm, which combine with each other to form little tapeworms. And these little tapeworms end up on the grass outside, assuming that feces is deposited in grass, which is a big if in North America, but pretty common in other parts of the world. A cow comes along eating the grass, and the cow eats these little tapeworm larvae. The tapeworm larvae move into the muscle of the cow. The cow is harvested for beef, and someone likes their steak fairly rare, and they don't like it. In other words, they don't like it cooked very thoroughly, and so they don't kill these little encysted larvae. They eat that beef, and inside the stomach of the person, this little encysted larvae hatches out and grabs onto the inside of the human small intestine and starts adding segments. And they can obtain lengths of up to 12 feet inside the human intestine. There's an X-ray that shows a tapeworm wound around, that's inside the small intestine there. So tapeworms are interesting creatures. They're, they are parasites that have relatively simple body shapes. They don't have eyes. They don't have skin. They don't have any of those things, but they don't really need those things because they reside inside a living organism